Welcome, everybody. Welcome to the FRAXA seminar series. I'm Dr. Mike Tranfaglia, and I'm the Chief Scientific Officer of FRAXA Research Foundation. And I just wanted to tell you a little bit about our webinar today. We were all very disappointed when all of the Fragile X meetings got canceled this year, for obvious reasons. And so in an effort to take their place and keep the information disseminating in the Fragile X field, we started this virtual seminar series. It's been very popular, and best of all, we'll be recording this for presentation on the FRAXA website. So I'd like to get started with today's seminar. We have a couple of senior investigators from one of the Fragile X centers who are going to be discussing the work that they're doing. We have Dr. Peng Jin and Dr. Zhexing Wen from Emory University. And Dr. Wen will be starting first. He's the Associate Director of Psychiatric Genetics and Epigenetics Core at Emory University. He received his PhD from Rutgers University in 2008. And he is going to be, let's see. Dr. Wen is going to be talking about the work that they're doing on organoids and figuring out the function of Fragile X protein. And so I'll turn it over to you, Dr. Wen.
Thank FRAXA to organizing this wonderful event, and thank Mike for the nice introduction. And today, I will talk about using human-induced pluripotent stem cell-derived 3D brain organoids to study the Fragile X syndrome. As we know that Fragile X syndrome is the most common inherited form of intellectual disability and a leading genetic cause of autism spectrum disorder. Fragile X affect both male and female. However, female often have milder symptoms than the males. And the exact number of people who have Fragile X is unknown. But studies show that estimate that about 1 in 4,000 males and about 1 in 6,000 to 8,000 females has been diagnosed with Fragile X. And the clinical features include intellectual disability, behavioral psychological abnormalities, and connective tissue dysplasia, and microautism. And in most known cases of Fragile X, the causative mutation is the trinucleotide CGG expansion in the fine-point UTR region of the Fragile X mental retardation gene called the FMR1. In humans, the number of the CGG repeats is highly polymorphic. Normal individuals have between 6 and 54 repeats, with 29 or 30 repeats being the most common allele. When the number repeats expand to between 60 and 200, it's referred to a premutation allele. When the repeat number reach over 200, it's known as the full mutation and leads to hypermethylation and epigenetic silencing of the FMR1 gene, resulting in the loss of the protein product called the Fragile X mental retardation protein, or FMRP, which in turn causes Fragile X. FMRP is a selective RNA binding protein found to have a major role in negatively regulating the translation of bound mRNA, especially at the synapse in neurons. Recent studies suggest that FMRP inhibits translation of its target mRNA by either equalizing the G-quant structure or G-rich region of the target mRNA, or binding specific RNA recognition elements such as ACUK and WGGA, or binding mRNA targets indirectly through either the small no-coding RNA brain cytoplasmic RNA1 or BC1, or the microRNAs. The function of FMRP and neuronal dysfunction in Fragile X has been extensively studied in animal models, including the fruit flies, zebrafish, and mouse models. The loss of FMRP leads to abnormal translation of selective mRNA, many of which are autism-linked genes. Substantial progress in the characterization of the underlying disease mechanisms in animal models has led to highly successful preclinical study of drugs moderating the mGluR pathway or the GABA receptors. However, follow-up clinical trial in human has been largely unsuccessful, and several challenging questions still remain unanswered. For example, what are the functional impact of FMRP deficiency in human neurodevelopment and functions? And what are there human-specific mRNA target of FMRP in human brain development and function? And what are they? And can we identify human-specific therapeutic targets and develop novel therapeutic strategy for Fragile X? To answer these questions, we need a complementary human-specific model. One approach is called the human-induced pluripotent stem cell, or iPS cells. This technology was first developed by Dr. Yamanaka in 2006, first in mouse, and then in 2007 in human. By introducing 4 transcription factors, now we call them as the Yamanaka factors, the skin fibroblasts could be regenerated back into the embryonic stem cell-like cells, and we call these cells as the induced pluripotent stem cell, or iPS cells. And the iPS cells have been shown to be very useful for disease modeling and drug developments. Basically, we could collect the somatic cells from the patients, including the skin biopsy, the blood samples, or urine samples. And then by introducing these 4 Yamanaka factors, we could reprogram these somatic cells into the iPS cells. And at the same time, we can also do the gene editing to correct the mutation in the patient cells, or introduce the same mutation into a healthy control background. And then we could further differentiate these iPS cells into the cell type that have disease relevance, for example, the neurons. And then from this patient neurons, we can study what happened in the patient neurons and what's the molecular and cellular phenotype there. And after we identify the phenotype, we could use those phenotypes as the readout to establish a high-throughput screening assay to screen compounds that can use back to the patient. And at the same time, the cell type differentiate from the iPS cells could also be useful for the future cell replacement therapy, for example, neurons for the self-replacement therapy for Parkinson's disease in the future. And compared to other model systems, the human iPSCs have several advantages. First, these patient-specific iPS cells carry the exactly same genetic information from the patients. This is extremely useful to study complex human disease, for example, those diseases that the animal model, it's very difficult to be made. And second, these iPS cells are multipotent. It means that potentially we could generate any cell type in our body from the iPS cells, especially those cell types have disease relevance. And the iPS cells are renewable, means that we have unlimited source for the disease modeling, for drug screening, and for the cell replacement therapy in the future. And it's easily accessible. We only need a small pieces of the skin biopsy or a small volume of the blood samples or urine samples. And there's no age limitation. We could generate the iPS cells from subjects at any age. And importantly, as I mentioned, that this is the patient-specific. So it's the individual-specific cells that could be very useful for the personalized medicine in the future. But also, at the same time, the human iPS cells also have several disadvantages. So first, we know that it's an in vitro system that we cannot do the behavior test in these cells, definitely. And also, compared to the cells in vivo, the cells differentiate from the iPS cells so far still quite young, and also lack of the myelination for the neurons differentiate from the iPS cells, and the neuronal circuitry also quite artificial in the in vitro system. So many differentiation protocol has been established by many groups. For example, right now we have been a different group have established the protocol to make different cell type in the human brain, including different type of neurons. For example, the cortical glutamatergic neurons, hippocampal neurons, GABAergic inhibitor neurons, and the dopaminergic neurons, as well as the motor neurons. At the same time, we can also generate the glia cells from the iPS cells, including the astrocyte and the microglia. With this iPSC-derived neuroprogenitor cells and cortical neurons, we have used this system to study the synaptic dysregulation caused by the mutation in one of the gene called the DISC1 gene in the human iPSC models of mental disorders. We also use this system to study the mechanisms underlying the microcephaly induced by the Zika virus infection. And we perform a repurposing screening to identify small compounds that can inhibit the virus infection or protect the neuronal cell death upon the Zika virus infection. However, we know that all these NPCs or neurons are grown in the 2D environments. And we know that the human brain is a 3D structure. And the 2D culture system, they lack brain cytoarchitecture and lack 3D microenvironment. And also in this culture system, the system makes many cell types. So to overcome this limitation, in 2013, Lancaster and colleagues had developed a protocol to generate a 3D structure with the traditional bioreactors. And this 3D structure called the brain organoids. And later on, when I was a postdoc in Dr. Guori Ming and Hongjin Song's lab, the student in the lab have developed a miniature bioreactor using the 3D printing and to increase the throughput and to grow the organoid and also reduce the viability of the culture system. And here is how the miniature bioreactor inside the incubator and the organoids in the traditional 12-well plate and how they look like. And furthermore, we modify the protocol to generate the brain region specific organoids. For example, here is the forebrain organoids, or called the cerebral organoids. And here is what they look like in the 12-well plate. And when we zoom in, we can see that inside the organoid, we can find similar structure to the human cortex, including different layers. For example, the ventricle zone-like layer, subventricle zone-like layer, and the cortical plate-like layer. And importantly, this forebrain organoid recapitulates human cortical development. For example, at the early time point, like the day 28, the majority structure inside the organoid are the SOX2 positive ventricle zone-like layer. And later on, at day 56, we can see that the neuroprogenitor cells from the SOX2 positive layer start to migrate out and differentiate to neurons and form the cortical plate layers. And even later, and at day 84, we can see that inside the cortical plate layer that we can form the different sublayers, including the CTIP2 positive, TBR1 positive tip layer, as well as the SEPI2 and CUS1 positive upper layer, similar to the structure of the human cortex. And importantly, this human brain organoid have a primary specific layer structure called the outer subventricular zone. That is the absent in the mouse models, and it's very important for the neurogenesis. And next, we want to know what's the age of the organoid we generate in a dish. So to do that, we compare the transcriptome profile of the organoid at different stage to the human fetal brain at different stage. And we can see that the early stage of organoid, like the day 26, is similar to the fetal brain before the 8th week, and the day 50 organoid similar to the fetal brain in the first trimester, and the day 100 old organoid similar to the fetal brain in the second trimester. And in addition to the excitatory glutamatergic neurons inside the organoid also contain other cell types, including the GABAergic interneurons as well as the exercise. And we have successfully used this forebrain organoid system to study the mechanisms underlying the microcephaly caused by the Zika virus infection. And we saw that Zika-infected brain organoid resemble key feature of microcephaly. So including the small size of the organoid, and it mainly target the Zika virus mainly target the NPC in the ventricular zone, and it caused the massive cell death and arrest the NPC proliferation. So to model the Fragile X syndromes and study the consequence of FMRP deficiency in the context of the neurodevelopments, Emory Fragile X Center, including Warren Lab, Basel Lab, and Gene Lab, and our lab, has generated iPS cell lines from skin fibroblasts of 3 Fragile X male patients, as well as the 3 age and sex match healthy controls. We also generate an isogenic correction line by deleting the expand CGG repeats in one of the Fragile X iPS cell line using the CRISPR-Cas9 gene editing. To determine whether loss of FMRP dysregulate human cortical development, we team up with Dr. Peng Jin's lab to and derive 3D forebrain organoids from the iPS cells using the established protocol with the miniature bioreactor I just talked about. And we focus on the day 56 forebrain organoid, which strongly correlated with human fetal brain development during the mid-fetal period, a critical period for human cortical neurogenesis and FMRP depletion in the Fragile X patients. We confirm the FMRP was absent in iPS cells embryo body at day 26, day 28, and day 56 forebrain organoids derived from Fragile X patient iPS cell lines. To examine the impact of FMRP loss on the neuroprogenitor cells or NPC proliferation, we co-immunostain Ki67 proliferation marker with SOX2 and NPC marker. We found a significant reduction of Ki67 positive cells among all the SOX2 cells within the ventricular zone-like structure, indicating a reduction of NPC proliferation rate at day 56. In addition, we expose control and Fragile X organoid to EDU for 24 hours, and then measure the cell cycle exit by assessing the co-localization of EDU with Ki67. And the Ki67 negative EDU positive cells indicate the cells that has exited the cell cycle within the 24 hours of EDU exposure. We found that the number of Ki67 negative EDU positive cells was significantly increased in the Fragile X organoid compared to the control at the day 56, suggesting accelerated neuroprogenitor cell cycle exit in the Fragile X organoid. NPC gave rise to TBR2 intermediate neuroprogenitor cells, or the iPSCs, which further differentiate into neurons. We found that while most TBR2 iPSCs are located at the edge of SOX2 positive ventricular zone-like structure in the control organoid, many of the TBR2 positive cells have migrated out to the CT2 positive layers in the Fragile X organoids, further suggest premature differentiation caused by the loss of FMRP. And we further assess the neuronal differentiation by quantifying the relative thickness of the substitute ventricular zone layer and the CTIP2 cortical plate layer between the apical and basal surface at the day 56. Quantitative analysis showed dramatically reduced ventricular zone layer thickness, while the cortical plate thickness was increased in the Fragile X organoids, likely due to the premature differentiation of the Fragile X NPCs. To confirm the neuronal fate after differentiation, we quantify the ratio of glutamatergic and the GABAergic neurons by co-immunostaining of chemokines 2, a marker for the glutamatergic neurons, and the GABAergic interneuron marker GABA. Interestingly, we found that the percentage of GABAergic interneurons was dramatically decreased in the Fragile X organoids, indicating a reduction of GABAergic neuronal fate. We further characterized the synapse formation and neuronal function in Fragile X forebrain organoid to investigate the impact of FMRP loss on synapse formation. We quantify the density of synaptic puncta by immunostaining of the synaptic vesicle protein called Zhexing Wen and dendritic marker MAP2. The density of Synapsin 1 positive synaptic boson was significantly increased in the Fragile X neurons compared to the control neurons in the day 56 forebrain organoid, suggesting an accelerated formation of morphological synapse by the Fragile X neurons. To further examine the physiological properties of these Fragile X neurons, we performed the electrophysiological whole cell petrogram recording in the slice acutely section from the organoids. And we confirmed the cell identity by injection of the Alexa fluoridine into the neurons after the recording and co-immunostaining of neuronal subtype specific markers, such as the CTIP2. And we found that the passive memory potential, including the passive memory properties, including the resting memory potential, the memory capacitance, and the input resistance, were not altered by the loss of FMRP expression. And we next examine the neuronal excitability under the current clamp mode with step of current injection. Interestingly, we observe a significant increase in axon potential firing frequency, suggesting the hyperexcitability of Fragile X neurons. We further characterize the detailed properties of the first axon potential. The quantitative analysis showed a significant reduction of the decay time of the first axon potential in Fragile X neurons, while the amplitude of the first axon potential, the half width of the first axon potential, and the threshold, as well as the rise time of the first axon potential, were comparable between the control and the Fragile X neurons. Furthermore, to figure out why the Fragile X neurons exhibit hyperexcitability, we measure the transient inward and sustained outward current density of the Fragile X and control neurons. And we found that the Fragile X neurons display larger voltage activated potassium currents, but normal voltage activated sodium current, suggesting an increased expression of potassium channel in the Fragile X neurons. Given the observed cellular and electrophysiological properties, phenotypes in the Fragile X organoids, we determine how the loss of the FMRP could impact the gene expression during the human brain development. We performed the bulk RNA sequencing using both control and Fragile X organoids at different developmental stages, including the day 28, day 56, and day 84, and identify a number of genes that are differentially expressed in the percent of FMRP in a developmental stage dependent manner. Given the significant number of genes with altered expression in the Fragile X organoids, we perform a similar RNA sequencing analysis using the mouse brain at the embryonic day 13.5, which is a similar developmental stage to a human day 56 organoid. Interestingly, we only observe 3 genes were differentially expressed in the absence of FMRP in mouse, and one of which is the FMR1 gene. While we found a significantly increased number of differentially expressed genes in the human forebrain organoids. We further examine the genes that display human-specific alternation in the absence of FMRP in forebrain organoid. These genes are associated with neurodevelopments, many of which has been implicated in the neuronal migration, axonogenesis, and the neurogenesis. Altogether, this data suggests that the loss of FMRP in human causes a more pervasive gene expression alternation during brain development compared to the mouse. However, the bulk RNA-seq is unable to provide detailed insights, such as the distinct cell population alternation, the presence of specific cell types, and developmental variation. To more comprehensively understand the specificity and the complexity of the impact of loss of FMRP on human brain development and function, we perform single-cell RNA sequencing on control and Fragile X forebrain organoids. Here we can see that the UMAP plot were visually adopted to identify 14 clusters as representing neuronal lineage based on the cell type specific molecular signatures in combination with the geo analysis. It shows that most of the cell population in an uncommitted state, such as early neuroprogenitor cells, the C2 here, and the radioglia neurostem cells, the C4. C4 is here, yes, and are greatly expand in the Fragile X organoids. Most importantly, the intermediate progenitor cells, the C10 population here, will not only increase in number, but also display a defined pattern of projection in the UMAP plot, creating a very narrow neck leading to the differentiated immature neuron assemblage. This marked change in this typically divergent projection close to the immature neurons suggests that Fragile X intermediate progenitor cells undergo distinct developmental progression compared to the control. And the DCX, the double clotting, is a marker of neuronal precursor and immature neurons is involved in neural migration. The DCX expression is abnormally stronger in the neuroprogenitor population, the C2, and the early inhibitor neuron, the C5, compared to other cluster in the Fragile X organoids. Interestingly, the more mature state neuron marker, tube 3.5, did not appear to be expressed in the C2 and C5 cells with high DCX expression in Fragile X organoids, suggesting that neurostem cells, progenitor cells of Fragile X organoids are primed to migrate prematurely due to the abnormal overexpression of DCX, which may lead to abnormal cortical layer formation in the Fragile X organoids. To further investigate the developmental progression alternation caused by the loss of FMRP, we utilize the statistically method based on the minimum spanning tree to obtain the pseudotime trajectory to investigate the developmental stage of cell population. So 14 pseudotime trajectory clusters were identified, and we locate the branch bypass breakpoints that were unique to the Fragile X trajectory, the blue dashed line, which was quite distinct from the control organoid time trajectory, the red dashed line. These cells, the cells of this subpopulation with all the neural state are strongly enriched in Fragile X organoids. The characteristic transcriptional feature of each branch cluster, the yellow mark, this yellow mark, define the differential dynamics of gene regulation in the developmental progression along the distinguishing developmental strategy. And this single cell RNA-seq data and the time trajectory analysis suggests that the loss of FMRP lead to alternative and differential developmental progression, possibly related to the aberrant premature differentiation of the Fragile X organoids. So I will stop here and come to the summary. So our studies show that human iPSC-derived brain organoid provide a unique platform for studying human brain development under normal and disease conditions. And the Fragile X4 brain organoid display human-specific phenotypes, including a reduced proliferation of neuroprogenitor cells, accelerated neural differentiation, and cortical layer formation, a deficit in the production of GABAergic interneurons, accelerated a synapse formation, and the neuronal hyperexcitability. And our bulk and single cell transcriptome analysis revealed that loss of FMRP could lead to pervasive gene expression alternation in the human brain development and altered developmental trajectory of the four brain organoids in a cell-time specific manner. So here I want to thank all the members in my lab, especially the people involved in this project in my lab and in Gene Lab. And I would also like to thank all our collaborators, including Hao, Gary, Steve at Emory, and Gordy Hongjin at UPenn, and Lu at Stanford. And I would also like to thank all the funding support, especially FRAXA provide the funding support to generate our important preliminary data for this project. Thank you for your attention.
Thank you, Dr. Wen. That was an excellent presentation and really fascinating results. I'm sure that will stimulate a lot of discussion and many questions. And I just wanted to note that we will have a question and answer session following the second presentation. But if you have any questions for Dr. Wen, you can get them in the queue now through the chat function. So you can start asking your questions and we'll take them in order as they come in. But we'll do that after the second presentation. So for our second presentation, we have Dr. Peng Jin, who is the chairman of the Department of Human Genetics at Emory. And for those of you who've been following the Fragile X field at all, you know that Emory has been a center of gravity for Fragile X research right from the beginning. Well, now it's also a newly funded Fragile X Center of Excellence via the NIH funding mechanism. And so Dr. Jin will present some of the work that they've done analyzing targets of FMRP in human tissues, and also talk about future directions for the Fragile X Center at Emory. I'm also pleased to say that Dr. Jin has been a many times FRAXA grant recipient, so we like to think that we've helped at least a little bit in the funding of their lab. So I'll turn it over to you, Dr. Jin.
Thanks, Mike. I have to say, from the day when I was a postdoc in Steve's lab, FRAXA has been providing a lot of support for my own research program as well as the training in my lab. And I just want to say thank you, Mike and Katie, and for your commitment and support over the years. As Mike mentioned, so I'm going to really just want to follow up on what Zhexing just discussed, to really dive in terms of molecular aspect of the FMRP in the context of human organoid. And then I think by the end, I'm going to spend some time to really give you some update in terms of what we plan to do as part of the NIH-funded FRAXA Center. So to discuss that, I have to say I have to step back a little bit, so everybody here is probably pretty familiar with epigenetics. So in Fragile syndrome, you have this expanded CGG repeat, and then hypermethylation and a loss of transcription of FMR1 gene, which leads to Fragile syndrome. But also, not only at the DNA level, in terms there is epigenetic modulation, there is also RNA level in terms of RNA modification, what we call either epitranscriptome or RNA epigenetics. So the story started really several years ago when we started the collaboration with Tranfaglia at U Chicago to study one of the most prevalent of RNA modification. It's called M6A. So over the last several years, it has been shown that M6A is dynamic and can be recognized by specific M6A binding protein, what we call M6A readers. And so those readers can regulate the M6A marked transcript in the context of the splicing, translation, mRNA stability, as well as the mRNA localization. So when we try to map the M6A profile during the neurodevelopment in mouse brain, and so we thought maybe we should really look at in the context of FMRP-mediated gene regulation. So shown here, I mean, we actually overlaid 3 data sets. One is in terms of the mRNA that could be marked by the M6A in mouse brain, and also which is indicated by the blue color. And the green one are the well-known about 800 FMRP clip gene done by Donnell's labs. And then on the bottom to the right is the differential gene expression that we detected using the same tissue. So shown here to the left is really just, we only highlight the significantly upregulated gene. As you can see, the number of overlap, the number of genes that could be marked by M6A and bound by the FMRP and upregulated in the absence of FMRP is very small, only 10 of them. However, if you really look at the genes that were downregulated in the absence of FMRP, the number that overlap among 3 groups significantly increased. So that actually gave us the first hint that the M6A could be involved in FMRP-mediated gene regulation. And so when you actually look at this very carefully, and the previous work done by Steve and Gary's lab, they show that they, by examining all the known FMRP targets, they identify the motif that are enriched among those targets. For example, like a GGAC or GACU and the GACUA. As you can see, actually, all of them are the known motif for the M6A modification as well. So that actually led us to think about maybe FMRP could utilize the M6A pathway to modulate the mRNA stability. And so then we decided to use a in vitro system to really test that idea. And so first of all, we use siRNA to knock down the FMR1 in the neural 2A cells, and then measure the mRNA stability. And as you can see, by inhibiting the transcription initiation through actimycin treatment, we can see that the loss of FMR1 can lead to increased instability of those FMRP mRNA targets. However, when we do the double RNA knockdown, which is both FMR1 and the YTHDF2, which is a known M6A reader that are involved in mRNA in regulating mRNA stability. As you can see, when we knock down both FMR1 and the YTHDF2, and we can actually restore the mRNA stability of those specific FMRP mRNA targets. So that indicates that FMRP could regulate the stability of specific FMRP target through the M6A and the mRNA degradation pathway. And so that led us to propose the following model. So in the mRNA that marked by the M6A, which can be recognized by the YTHDF2, and then which can recognize both YTHDF2 and the FMRP, and the FMRP could potentially protect those mRNA from being recognized by YTHDF2 and then further mRNA degradation. So again, this really is just a biochemical association, and it's an in vitro analysis. So to further to test their physiological relevance, so we decided to utilize the fly model that we established previously, and so to simply to cross the FMR1 mutant fly with the M6A mutant line. And so we utilize two phenotypes, two well-known phenotypes that are associated with FMR1 mutant. The first thing is the neuromuscular junction overgrowth phenotype. And so I think many of us here know that when you don't have the FMRP in the fly, you will lead to the synaptic overgrowth in the neuromuscular junction. And so when we cross the FMR1 mutant line with the RNA knockdown line of the METL14, which are the key enzyme for M6A modification, you can see there is strong genetic interaction, further enhanced and the synaptic overgrowth. So that indicates there is a pretty good genetic interaction between FMR1 gene and M6A pathway. Furthermore, we also look at this at the behavior level. And again, we utilize well-established causal behavior phenotype associated with the FMR1 mutant. As you can see, again, that loss of the METL14 in the FMR1 null background, we can further elevate it, enhance this the causal behavior deficit associated with the loss of FMR1. Besides our work, there's also additional work from Chuan He's lab as well as Hongjun Song's lab and Yung Chao's math lab. They have shown that, besides regulating the M6A mediated mRNA stability, FMRP could also potentially utilize M6A pathway to regulate the nuclear export as well. So why I was talking about that? So in the meantime, we were also part of the Psychiatric consortium. So to map differential M6A modification during the human brain development. So in collaboration with Hongjun at Hopkins at that time, and so we actually did this kind of comparative analysis between the human fetal brain and the mouse fetal brain. And so what struck us the most is that there's really pervasive M6A modification in human tissues, as shown here. And then all the blue dot indicate that those mRNA that specifically marked by M6A only in human brain, and so the yellow-green ones are marked by both M6A marked by M6A in both human and the mouse. So, and also the genes that are specifically marked by M6A in human have been shown related to mental disorders, intellectual disabilities, as well as neurodegenerative disorders. So that actually, this was done back in 2016. So that's actually one of the reasons why we decided to team up with the Zhexing lab to really look at the role of FMRP in human brain development in the context of human brain organoid. So as Zhexing has presented, there's a pretty consistent striking phenotype associated with the loss of FMRP in the human brain organoid. So in the meantime, we decided to, given what we saw with M6A distribution in human and the mouse brain. So we decided to perform this parallel clip assay using both human organoid as well as the similar development stage of mouse fetal brains. And so we performed the e-clip assay using the FMRP specific antibody. And so this is the summary data from multiple e-clips analysis. So first of all, what we found is there is significant overlap between the human and the mouse e-clip data sets. There are about 1,600 mRNA that can be bound by the FMRP in both human and the mouse fetal brain, as well as human organoid. And so among those 1,600 mRNA, 80% of them actually, those 1,600 overlap with 80% of the FMRP target identified by Daniel's group. So that indicate we can really reproduce many known FMRP mRNA interactions. Interestingly, when we compare human and mouse e-clipped mRNAs, what we found is that there are many more mRNA that could be bound by the human FMRP, but not in the mouse, but not by the mouse FMRP. Further gene ontology analysis, and so we look at what genes, what pathway are enriched among the genes that overlap between human and the mouse, and among the human specific, as well as the mouse specific. Interestingly, I think if you look at overlapped mRNA target, a lot of them involve in the synaptic plasticity, synapse assembly, as well as IGF receptor signaling pathways. So all those pathways are well known involved in the FMRP-mediated gene regulation. Intriguingly, when we look at those mouse-specific FMRP mRNA target, the gene that really on the top is glutamate receptor signaling pathways. And so that indicate, it seems like there are more a gene involved in glutamate receptor pathway could be bound by the FMRP specifically in the mouse brain. So due to the time limitation, I'm really going to focus on one of the human-specific mRNA targets that we identified, which is a CHD2. So CHD2 is a gene known that involved in intellectual disability and epilepsies, and has been shown to be one of the 6 strongest autism genes. And so it has been shown that CHD2 is critical for neurogenesis and the GABAergic neuron generation. And so the loss of CHD2 could lead to the aberrant cortical network function and impaired the memory. To really confirm that this FMRP, only human FMRP combined to the CHD mRNA, so we performed the traditional FMRP immunoprecipitation experiment, followed by the real-time quantitative RT-PCR. And so what we found confirmed is indeed only human FMRP combined to the CHD2 mRNA. Furthermore, we want to look at in terms of both mRNA and the protein level. So what we found is at mRNA level, there is no change in either FMR1 knockout mice or Fragile X organoid. However, we did see the CHD protein level increase only in the human brain organoid. So that indicates that CHD mRNA could only be regulated by FMRP in the human organoid. Furthermore, since CHD2 is a known chromatin modeler, and so we want to see what's kind of downstream gene that could be regulated by the CHD2 in the absence of FMRP. As shown here in this pie chart, you can see all the orange fraction. In total, there are about 408 genes that display differential expression in FRAXA organoids, and that among the genes that are regulated by the CHD2. Interestingly, among those 408 genes, there are about 255 genes that could be bound by the CHD2 as a promoter region. So again, I think that indicates, when you have this upregulation of the CHD2 in the absence of FMRP, and that can have direct impact on the transcriptional level of those CHD2 or non-CHD2 mRNA target. So in summary, I think for this section, for this part of my talk, I hope that I have convinced you, besides the DNA modification, that there are actually many more RNA modification in the genome, and then the RNA modification could be more pervasive in human brain. So that's another reason why we need to really look at the human brain development in the context of in the context of RNA modification, as well as the neural development. So our data our eClip analysis have also identified some human-specific mRNA target. We believe those targets could potentially serve as a therapeutic target for the Fragile syndrome. Also, I just want to thank people in my lab and also my longtime collaborator, Chuan-Hee at Chicago, and Hongjun Song from Penn, and David Nielsen from Baylor on Fragile pre-mutation. As Mike mentioned, my mentor, Steve Warren, and also all the colleagues with our previous Emory National Fragile Research Center here. Also, as well as all those funding agencies, again, particularly for FRAXA Research Foundation. I think without Mike and Kitty's support, I think we would not have been able to really start the organoid project. So I will, what I want to do next in the next 10 minutes or 15 minutes, if you allow me, to really give you some update in terms our recently founded Emory National Fragile Research Center. So the PI of the center is me and Dr. Warren. And so we have actually assembled a team from Emory, NYU, and Stanford, Penn, as well as USC, to really want to take very systematic approach to really utilize the human brain organoid to understand the role of FMRP mediated gene regulation, and potentially based on our funding using the organoid, we can identify new therapeutic targets. So as Zhexing already indicated, I think this the iPS cells on both 2D and 3D culture system could really help us to understand the human brain development, and so which has been a very important platform to really model the human brain development and study the disease mechanism. And in the context of Fragile syndrome, I think little is already known on FMRP-mediated regulation in human brain development. So we feel this can be a great opportunity for us, as a group investigators, with a different expertise to really address this question together. So our overall hypothesis is that FMRP could regulate human brain development and circuit function, where the regulation of novel mRNA target, which could serve as a new therapeutic target for Fragile syndrome. And so our proposed center will really integrate different type of analysis from the transcriptome, genome-wide translational profiling, single-cell genomics, or cell type-specific manipulation, as well as high-throughput small molecule screening. So we have three center specific aims, which were really have a lot of crosstalks across three different projects, which I'm going to get to in the next slide. In the first aim, we're going to determine the role of FMRP in human brain function. And in the aim 2, we're going to systematically identify the functional mRNA target of FMRP in human brain and to understand those mRNA target in normal brain function. And third is to really develop the novel therapeutic strategy for Fragile syndrome using the organoid as a platform. So here is our team. And so in the project 1, we are going to, which will be led by Gary Bassell at Emory and Eric Klein from NYU. And in the project 2, we're going to be led by Lu Chen and Tom Sudoff from Stanford. And then for project 3, which will be led by Zhexing Wen, Steve, and me at Emory. So in the project 1, so this particular project, we're going to focus on the mechanism and the function of FMRP in iPSC-derived system to study the human neural brain development. So the rationale is that, as Zhexing has indicated earlier, although despite all the successful preclinical study conducted in animal models, the clinical trial in FedEx has failed so far to really meet their defined primary endpoint. And so I think there could be a possibility that there could be, unknown human FMRP biology, which really require further investigation. And also, as Zhexing demonstrated, and also the work done in Gary Bassell's lab using the 2D culture system. And so we have also identified multiple phenotypes associated with those iPSC-derived model system for Fragile syndrome. So in terms of the aim, so for this project, Gary and Eric were going to trying to characterize the cellular phenotype across the human brain development caused by the loss of FMRP, and identify the dysregulated FMRP target that could potentially drive the phenotype that we have observed in both 2D and 3D culture. And then the last experiment is really to do the functional rescue experiment, and by utilizing the identified human FMRP mRNA target. They are going to utilize really multiple different approaches, and also multiple comparisons in terms of MBC versus neuron, etc., versus inhibitor neurons, and as well as like a different molecular technology such as single cell RNA-seq and ribosome profiling. In the project 2, Lu Chen from Stanford and Tom Sudoff, also from Stanford. To some of you who don't know, and Tom received the Nobel Prize back in 2013. I know Mike and Katie had been very successful to recruit Nobel laureates to study Fragile syndrome. I think this is the third one, I believe. And I think for this project 2, Lu and Tom going to really characterize the synaptic phenotypes in both human Fragile organoid and mouse models. And I think the goal here, as Zhexing indicated earlier, I think one of the disadvantage using the using the organoid as a model system is that we cannot really look at the in vivo function, particularly like electrophysiology and the learning and memory. So Lu and Tom actually are going to take advantage of both systems, both the FMR1 knockout mice and the FedEx organoid model that we are generating, and to really try to examine both convergent and divergent mechanism underlying the FMRP-mediated regulation in the context of the cognitive impairment associated with the Fragile X syndrome. So in their project, there are two aims. The first, they are going to really, again, the first one, in the aim one, they're going to focus on the Fragile X organoid system that we developed to determine both neuronal and synaptic phenotype associated with the loss of FMRP. And in the aim two, they are going to investigate this mechanism underlying the cognitive deficit associated with Fragile X syndrome using the mouse model. I think there will be a lot of crosstalk between the human Fragile X organoid and the Fragile X mouse models that we have. And last, for the last project, so Zhexing, Steve, and me, we're going to really drive this direction, trying to develop the therapeutic strategy using both iPSC-derived neuronal cells as well as the 3D brain organoid system. So we have actually three directions. So we really want to do this systematically. In the aim one, we are going to really determine the therapeutic effect of all the known compounds from the, let's for example, like a mGlu5 antagonist to GABA agonist, and as well as metformin and the S6K inhibitor as well in the context using the Fragile X organoid as a model system. In the aim two, we're also going to test and compare the CRISPR-based genomic and epigenomic editing strategy using Fragile X organoid to see which one would be more efficient and what the reactivation is really needed to be able to rescue the phenotype associated with the Fragile X organoid. And last, I think is more open-ended. So we are going to collaborate with NCATS, the part of the NIH Institute to really to perform this molecular phenotype-driven screening. And so we are going to measure the gene expression of hundreds of genes that are altered in Fragile X organoid, and then screen probably up to 1 million compounds and trying to identify the compounds that could rescue suppress the phenotype associated with the Fragile X organoid. And then further coming all those strategy that we identified here will actually go back to the in vivo model as I discussed for the project 2 to be further tested. This is my last slide. And so I think the overall goal of our center is hope that we can really continue, as Mike mentioned, the tradition at Emory, and also with the new collaborators to gain additional insight into the core mechanism of FMRP-mediated gene regulation in human brain development. And hopefully we can review the convergent and divergent abnormalities associated with the loss of FMRP. We can identify species-specific and shared FMRP-mRNA target in both human and the mouse. And I think as a whole, I think if you really look at our 3 projects, our hope is really we can connect the dot from the molecular, cellular, synaptic to cognitive deficit associated with the Fragile X syndrome. And the last but not least is really, I think that's the ultimate goal, is really to identify the novel strategy for the therapeutic intervention of Fragile X syndrome. So I will stop here, and I think Zhexing and I will be happy to take your questions. Thank you.
Thank you, Peng. That was a really wonderful presentation and a really remarkable team you have assembled there, I must say. That's very impressive.
Thank you.
So we'll start with some questions that we have from Milos Kostic. Is the single-cell RNA-seq analysis in the single-cell RNA-seq, did you see heterogeneity in transcriptional profiles between control and disease lines? And I think that was for Dr. Wen.
Peng, do you want to address this question? I think our data is quite consistent with the data from Arata group. That overall they are large reproducibility of the organoids. But do you want to address more?
Yeah, I want to follow up on that. We do see heterogeneity in terms of single cell RNA-seq profiles, but I think what we are focusing on really the gene that, or cell type specific or consistent alterations across. One is multiple different control and disease lines, and also we have also done the single cell RNA-seq on the isogenic line that we have produced as well.
One follow-up question I had, or just a comment, is that traditionally we had thought of Fragile X as a disease of translation, and more now that we're identifying transcription factors among the mistranslated proteins, it's becoming also a disease of transcription as well. Bob Darnell, I think, reported a couple of years back on a transcription factor that was one of the mistranslated proteins. Mm-hmm. So anybody have a comment on that? To what extent have we really overlooked this as a major disease mechanism if we're focusing so intently on translation and ignoring transcription?
I just want to follow up on that. I think, with our study on the M6A pathway, I do feel, as well as Bob Darnell's study, I do feel like we need to look at both sides, and both transcriptional level as well as the translational level. And if you really look at as I mentioned, the CHT2 study, it seems like a lot of transcriptional regulation could be related to the CHT2, at least at that development point. So I think we need to look at both of them very carefully and together.
Are there others out there? Have we identified all of the transcription factors that may be translated abnormally?
No, I think Bob's one, that's another one. I think that's also an important thing to really for us to go back to the mRNA target and trying to identify additional ones and then look at them more carefully.
Okay, from Anita Bhattacharya. Hi, Anita. Beautiful talk, Zhexing. Do you have data, or is there data from fetal human Fragile X brain or even mouse models showing that there's a difference in lamination or migration of cortical neurons to in Fragile X to validate your iPSC organoid data?
Thank you, Anita. So we did do the RNA sequencing for the human fetal brain, Fragile X brain, and compare the transcriptome of the human Fragile X fetal brain and the Fragile X organoids. And we do see some overlap between the organoid and the fetal brain, especially those genes involved in the neurodevelopment. But because we only have very limited brain tissue, that we were not able to do the immunostaining to look at more detail of the cellular phenotype, unfortunately.
Yeah, also I want to follow up on that. So I think it has been difficult to really get the Fragile X fetal brain tissues. So one of my former postdoc, so she was able to get hold a couple Fragile X fetal brain tissues, which were like 10 years old in the freezer in the liquid nitrogen. And so we were able to get some RNA, get some RNA sample, get RNA out of those tissues and did RNA-seq So we do have those data. So those data will be published soon, and I hope those data would be useful to the community. And also when we overlap with the organoid data, they're about I would say probably depending on the development stage, there could be up to 30 to 40% overlap. So those fetal brain were 22, 23 weeks old post conception.
Okay, and from Hanggu Lee, another FRAXA fellow. Hi, Hanggu. Thanks for the amazing talks. And a question for Dr. Wen. Is it possible to test the EI balance in organoids? And for Dr. Jin, was the G quadruplex motif enriched in the ECLIP targets?
Yep, testing the EI imbalance is our next step for the electrophysiological study in the fragile X organoids. We will do that, but right now we don't have data for that yet.
For me, I think, Honggo, I think that's a great question. That's something we are still looking at them right now. So to really look for the enrichment of the G quadruplex motif. So we're still working on that.
Okay, from L. Barrett, for the human organoid ECLIP and mouse brain ECLIP comparisons, how similar were the peak locations between species and between binding motifs? What binding motifs?
Yeah, that's an excellent question. And so for the binding motif, it's pretty similar to what we saw from the mouse data that I mentioned earlier. In terms of peak location, so we have not compared that, and that could be there could be some computational challenge. I mean, if you consider human and mouse genes, so we have not really looked at that carefully. But if you have some idea, then we would be more than happy to chat with you.
Okay, and David Walker, pharma partner. Hi, David. For Peng, you mentioned testing S6 kinase inhibitors in organoids. Do you anticipate publishing the data?
Not yet. So that's something we are doing right now. Of course, if once we complete the study, we will certainly publish that.
I think he's looking for a sneak peek at the data. That's what he wants.
Nice.
Okay. Another question from Milos for Dr. Wen. Can you comment on the number of organoids that went into the single cell RNA-seq analysis, and how old were the organoids?
So we totally did single cell RNA-seq for 6 organoids, including 3 fragile X organoids and 2 control and 1 isogenic control. So totally 6 organoids, and we did all the single cell RNA-seq at the day 56 to be consistent with our cellular phenotype.
Okay. And I think that may be the end of the questions. Oh, there we go.
Hi. Hey,
one more from Pietro. Hi, Pietro. Long time no see. Let's see. Pietro wants to know. I think Pietro wants us to put in a little plug for the international workshop on Fragile X in Atlanta. When is that, Pietro?
So I can answer for him. So I volunteered I believe last year in Italy and to host next the X-linked MR meeting in Atlanta next year. So that's something I think we can certainly discuss what's the best way to really plan that meeting given COVID situation.
Yeah, normally they have these meetings in wonderful resort locations, which are usually a little bit too far away for us to make it to easily. But Atlanta.
Yeah, it will be not far away from Atlanta downtown. So it's there. We have a lake there, called Lake Lanier. So it's a beautiful place to be. At least that's what I'm thinking. But again, I just, with all the travel restriction, I just don't know how feasible we are doing the planning at this moment.
Well, here's hoping that we get back to some real meetings someday. But in the meantime, this was wonderful. And I really want to thank our speakers for today, for Dr. Wen, Dr. Jin. Fantastic job. And this will be accessible through the FRAXA website, the recording of this presentation. We thank you so much for being here, and keep up the great work.
Okay. Thank you, Mike. Thank you, Katie. Yeah.
Thank you
for having us. Bye, everybody.
Bye.
Bye-bye.